when basically also biology becomes physics, and you can do a theory, verify the experiment, and get the information, go back and forth. But I hope it will be apparent at the end of this session. So the first speaker is Sara Fortuna, computational design of peptide-based architecture for biomarker recognition. She's the coordinator of the section of molecular nanotechnology for the science application theory group. Sara.
So besides thanking the organizer for the opportunity of presenting my work in this beautiful summit, my name is Sara Fortuna, and I represent the Molecular Nanotechnologies for Life Science Application Theory Group. This does not. Oh. And the University of Udine, in short, MONALISA. Mission of our group is to provide novel nanotechnological solutions to problems of medical interest, and in particular, we aim to develop new nanodevices for separation, labeling, and detection. And in particular, we focus on the probe. The probe is this little sensing element capable of capturing in solution a molecule of interest. This could be, for instance, a biomarker in any biological fluid. And in particular, we want to use the computational tools to answer a number of questions, such as, is it possible to design binders for a surface-exposed binding site, which is this kind of difficult problem? Can we use the in silico design binders to, for instance, immobilize proteins on surfaces? And is it possible to increase the binding by coupling two or more binders together? Stepping back, we all know that typical probes for protein recognition are antibodies. These are highly versatile macromolecules, and they are used routinely in standard tests such as immunohistochemistry or ELISA tests. They are produced in vivo and from cell cultures, which makes their production quite time-consuming and expensive. The problem with these methods for their development is that it's not easy to control which epitope our antibody will bind. Into more details, antibodies are large macromolecules composed by two heavy chains, here in green, and two light chains in blue. Where the light chains and the heavy chains meet together, we have the so-called antigen binding site. On the right of this slide, you see a zoom of the antigen binding site, the hypervariable region binding a protein, in this case lysozyme. You can see the antibodies is binding its target protein through a number of loops. These generally are a number of hairpins loops and two larger loops that we can think about them as peptides. We said that the antibodies developed in vivo or in vitro, for them it's difficult to pinpoint a binding site or an epitope of interest. To ease this task, is it possible to in silico design a binder targeting a precise binding site? And this can be done by in silico evolution. This is what we do in our group. The applications, for instance, can be to discern this among protein isoforms, and this can be useful in diagnostics or prognosis. For instance, two proteins isoforms can live in the same biological fluid. This is the case of HER2 protein, for instance, for the breast cancer. Another application could be to anchor a particular site of a cell membrane for drug delivery. It would be very nice to be able to evolve in silico a full antibody. However, antibodies are very large macromolecules. They are composed by 1,300 amino acids, and this means that from a computational point of view, they are really computationally demanding. They require a lot of computer power in the form of both memory and CPU time. However, peptides, which we've seen in the previous slides, are really the binding elements of the antibody. They are composed only by 10, 20 amino acids. This is the computational load and allow us to evolve them in silico. Our protocol is pretty simple. To get started, we need a target crystal structure. This can be done, this can be built, for instance, also by homology modeling. And given the crystal structure, we can choose on this crystal structure a binding site of choice, an epitope. For instance, if we want to compare two mutants, we could choose the mutation point or some structural modification on the protein surface driven by the mutation itself, something to differentiate the two proteins. Once we have the protein and a binding site, we need to choose a binder. This can be a peptide or a larger probe. And we need to choose which part of this binder we want to optimize. Once we have this element, we can proceed with the in silico evolution of our binders, which will generate a number of candidates that then will undergo in silico screening. This to solve some very practical problems, such as the solubility of the molecule we have designed, and to test its thermodynamic stability. Few candidates are selected by this screening, and they can undergo experimental screening to see whether actually it's possible to synthesize them or produce them, and of course to gather some preliminary result to choose the lead binder, which then will undergo full characterization. The in silico evolution of amino acid-based binders is based on a very simple iterative algorithm. Typically, we take a random peptide, say a polyalanine, we choose an amino acid at random, and we perform successive mutations. The mutated peptide, the mutated probe, is then tested for its binding affinity to a target protein. And the mutation is accepted or rejected with a certain probability. And here on the right, you can see a typical output. On the y-axis, we have the binding energy of the probe with the target protein, and on the x-axis, the number of mutations. And we'll see that as the evolution proceeds, the binding energy drops. Also, here there are a few simulation snapshots. Here in the bottom, there is the beta-2 microglobulin chosen as target, and cyclic peptide, again a polyalanine, cyclicized with a cis-cis bridge, put close to the binding site. As the simulation proceeds, the peptide gets closer to the protein surface, and its binding affinity becomes much favorable. From this run, we select a certain number of peptides. We run molecular dynamics simulations. This means we follow the protein binder evolution along time, and we make sure that the binding affinity, the distance of the peptide to its binding site, and its shape remain unchanged along the simulation time, in this case, 50 nanoseconds. The binders then go to experimental screening. For instance, in this case, is an SPR screening to make sure that the SPR signal is proportional to the peptide concentration. Next, we characterize the chosen peptide, and in particular, we check that it actually binds to the correct binding site. Here, for instance, in the top, we have the NMR result. Highlighted are the amino acids that are affected by the binding, and they are all on the proper side of the protein, proving that the binding site is the correct one. In the bottom, instead, we measure the height of a chip coated with a peptide before and after the capture of the beta-2 microglobulin. In red, the red line is the height of the system before the beta-2 microglobulin, in blue after. Their difference in height is about 2 nanometers, which correspond to the lateral extension of the protein, as it is bound to the peptide through this side. So it stays on the side. So the algorithm works. We can capture protein from solution, and we can immobilize them oriented on a surface. Current work is about optimizing nanobodies, which are small antibodies, and we guarantee a better binding. Another interesting problem, given that the peptides generally give a binding affinity in the micromolar range, the question is it possible to improve their binding by coupling 2 or more peptides, 2 or more probes? And this is exactly what nature does in the antibodies. as we've seen before, antibodies can bind their target not through only one loop, but through multiple loops together. We model this to see whether it's true or not that the chelating effect or linking to peptides is effective with a toy model that we call nanoheadphones. Here, these green spheres represent our biomolecule, our target, the biomarker we want to capture in solution, and the little chain and the chain of yellow beads represent a linker. You can think about it as a PEG chain or any polymeric chain of choice. The polymer can capture the protein through its end beads, and its size is close enough to the protein size. By running a number of simulations, we can measure the detaching free energy of our nanoheadphone. This is related to the binding affinity that you can measure in the lab. Here, in the bottom line shows the detachment-free energy of a polymer binding only through one end. The second line represents two equal binders binding only through one end, and all the other lines above this show the detachment-free energy of double binders. In all these cases, the binding free energy of detaching a double binders is higher. This tells us that actually it's almost always convenient to link two binders together to achieve better binding affinity. And in particular, in this two-dimensional plot, where we have the binding angle with respect to the number of beads of our linker, and the color code darker means higher binding free energy, tell us that a binding angle between 60 and 80 degrees and a chain length not too long, but long enough to guarantee the chelation of our protein is the best conformation. Now we are testing this in the lab, again with a beta-2 microglobulin and 2 peptides. These are preliminary results showing that the 2 independent peptides actually can bind the protein, and we are very curious to see if this is the case also by linking them together. In conclusion, the computational design, I'll show you that it allows to correctly target a chosen binding site of a protein, being that a pocket, results that I did not show you, but you can find in the literature, or our surface-exposed non-druggable sites, which is the difficult case, given that we use the tools for molecular docking. This is a very advantageous feature that can be exploited for oriented protein immobilization, and the ex novo optimization of peptides, starting from a random peptide, can reach micromolar binding affinity also for surface-exposed binding site. And the binding is supposed to be boosted by coupling two peptides together. And of course, current work is about nanobody optimization. Stay tuned on that. Thank you very much.
Thank you. The paper is open for discussion, questions, remarks. There must be a curiosity. Yes. Oh, just a moment. You need to speak on the microphone.
Okay. So could you explain again how many of these peptides did you screen with this mutation study, and I mean in silico? And was it iterative, or did you do it just once?
So we optimize one peptide, and from this optimization run, generally we choose between 10 and 20 peptides. On this, we run molecular dynamic simulations, and we check their solubility. And we generally end up with 10 peptides. And they went experimental screening in this particular work, but now we are even able to pick 1 or 2 peptides from the molecular dynamic run, and we are sure that with a good possibility, they are good binders. So we are close to arrive only to one candidate that is likely to be a good binder, which is an achievement.